Hi everyone, my name is Sarah Taffel, and today we are doing another episode of UFDPM's Program Research Talks. Today we have Zachary Eldred as our guest. So Zachary, if you could start by introducing yourself today.
Yeah, my name is Zach Eldred. I'm originally from Morgantown, West Virginia. I've spent the last about 10 years or so in Guam when I was in the Navy. And then I went to the University of Guam, and that's where I got my undergrad until I came here for the DPM program last August. So I'm currently in my second year of the DPM program.
So many DPM students participate in research throughout the program. So what is your field of research and why have you chosen this area?
So thank you. My field of research right now is involved in plant pathogens, and I'm currently researching pathogens in organic high tunnel production systems. I didn't necessarily choose this, but because the DPM program has so many different departments, and we get the chance to go and work in different departments, I was able to come here, and since starting here, I really enjoyed that.
So what were your methods in terms of collecting your data and how long do you think it's going to take?
So my data collection is based on scouting. So each week I go to the field and I scout for potential pathogens. I collect samples and then I bring them back to the lab and I grow out anything that we might want to look at. Also, I take a lot of soil samples because one of the big problems in high tunnels is fusarium. And so we take soil samples, and then we grow out seedlings in that soil as a bioassay and try to see if we can't cultivate any fusarium in the soil from the soil, which will allow us to know whether we have the pathogen in our high tunnel or not. And this can inform our grower what to do. I go to the field about once a week to take those samples, and then so each bioassay takes about 8 weeks or so from start to finish. And then after I do the bioassay, I look at the symptoms of the plant, and I'll then take the plant and cut out the roots or cut out the crown, and I also plate that, which takes about another week. And I just keep cycling these through each season, probably 3 to 4 times a year. I take different soil samples and go through the process of doing bioassays. Uh, and throughout the year, we're continuing different crops. So I'm always maintaining time in the field, doing different scouting and different crops, such as bok choy or spinach. And then we even through cover crops.
So far, what are the results that you're able to collect?
Yeah, so each year we've been seeing different pathogens. Last year we saw powdery mildew, and then this year we saw a lot more gray mold or passiflora. So it's hard to say what the results are because this research is still ongoing. But I have not seen any fusarium in our soil yet. We do have other pathogens such as southern blight, Sclerotium ralphsii. And really, our data is more of a presence or not present collection. We aren't running any statistical data. We're looking for pathogens in the high tunnel so we can make decisions in the future and trying to get an understanding of the high tunnel system in this growing environment, which is Florida. So far, our results have been good. We have collected many pathogens throughout the last 2 years of the project, but we have yet to be able to collect any fusarium, which is good for the grower. And we are continuing to learn more about organic high tunnel growing and pathogens and how to manage them in the environment. But this research, like I said, it's ongoing. So we're continuing to find more information and develop new data.
So you mentioned before that it can help growers in the field. So how can this help them specifically?
So one of the big aspects of this project, because it's a very large, it's very large. It's multi-state, multi-university. So the main goal is to be able to give information to the growers on what to expect, how to manage these problems, and we do that a lot through outreach as well. So growers who are interested in growing high tunnels, they can know throughout the year what to expect, and when they encounter certain pests or diseases, how they'll be able to go about managing those problems.
Well, thank you for taking the time to explain your research. It's clear that you have put a lot of time in this project. So, do you believe that the DPM program has prepared you well in order to take on this project?
Yes, I think the DPM program is really beneficial for this kind of research. I'm not the only DPM student working on this project. We have other students who are working in the insects, and uh, there's so many aspects to a large project that the DPM program, because it's so broad, allows you to look at many aspects of it and see it from multiple angles, which makes it beneficial to you doing your research.
And to finish up, do you have any advice for new DPM students on how to be successful when completing their research?
Yeah, I would say just take initiative, be inquisitive, look for problems, try to identify solutions, use the people you work with in the other departments, and see if you can make any connections. There's always places to grow, and there's so much knowledge out there and information for us to learn from in these projects, that making sure we reach out to those other counterparts can really be beneficial in seeing how to improve even your part of the research.
So, thank you very much, Zach. I appreciate you zooming in to share all this information to our viewers. And thank you to our audience for coming to watch another episode. Stay tuned for more.